Hello, everybody, and welcome back to BioSC 140 Human Physiology. This video is going to cover the experimental procedures and the discussion for laboratory 17 and 18. So there's two labs within this lab. There's the Biopack Lesson 3 Electroencephalography and Fox 3.3 reflexes, where we're going to explore the knee-jerk reflex, aka patellar reflex, know both names, and the plantar reflex. So let's get into it. The EEG, aka the electroencephalogram, know both names. Purpose: how does brain activity change during different activity levels? We're going to measure the electrical activity, aka nerve impulses, of the cerebral cortex. So in class, it actually kind of looks like this picture up here. So if you've ever seen a movie where somebody has all these like electrodes or pads all over their head, that's what we're doing in this lab. We're putting, it's similar to the electromyogram, where we put the electropads on our forearms and measured electrical activity in our forearms. Just this time we're doing it for our brain. And you know, people tend to have hair. I don't, on top of my head, but most of my students do. And so it can be a little difficult to get the pads to stick on people's heads. So that's why there's like this sheet wrapped around this person's head to help hold those pads on to get a better electrical reading through their hair. So instructions, procedure. Follow instructions in the supplement for Biopack Lesson 3. You set up the Biopack, where the Biopack is the computer system that we would use in a live lab. There's a laptop, and then there's a separate like data gathering processing unit that you connect to the laptop, and then that separate data processing connecting unit we attach the electroencephalogram to. So first you need to calibrate. So you set it up and then you calibrate. Calibration is you just kind of do like a trial run and make sure everything's working correctly. And then we're going to record the electrical activity of people's brains in 4 different situations. So first, the subject is going to be relaxed and have his eyes closed for 10 to 15 seconds. So close your eyes, relax. And we're going to see what his brain activity looks like at that time. Then they're going to open their eyes for 10 to 15 seconds. Then they're going to close their eyes again for 10 to 15 seconds. Then they're going to do the Gendrastic maneuver. The Gendrastic maneuver is where you kind of clasp hands, you grab your 2 hands together, and you hold on tight and you pull apart. So your hands stay together, but your muscles are activating and trying to pull apart. You're generating force, pulling your hands apart, but you're still kind of hanging on for 10 to 15 seconds. We're then going to analyze the data. We're going to look at standard deviation. We're going to look at frequency, and we're going to look at amplitude of different waves. So frequency is measured in hertz, and we're going to measure from peak to peak. So we're going to have waves. We're going to have data that's kind of going up and down, up and down, up and down in a rhythmic manner. We want to know how often those cycle. So one cycle would be peak to peak. So up and down, up and down. How long does it take to go up, down, then up? So start at the up, go down, and then back up to the side. That'd be one peak to peak. We're going to measure that for alpha waves, beta waves, delta waves, and theta waves. And we're also going to measure altitude. Altitude is how high is the top and how low is the low during one cycle. Perfect. So our data is going to come out looking like this. Our data is going to come out looking something like this. Alpha, beta, theta, and delta waves. have different frequencies and different amplitudes, and they will change throughout our different activities while we're recording. Box 3.3 is the Reflex Lab, and this is actually one of my favorite. I think this is a really cool thing to do in class, and I'm sad we can't do it live. But purpose, what is the physiological basis for reflexes? So the demonstrations in class are really cool. I'm gonna put some videos online so you can see them happening and see what it looks like. But it's really cool to do it in person, and we actually do it on each other, and I actually go through a whole bunch of reflexes when we do this in person. We do this one, the patellar, we do the plantar reflex, and then we also do like the Achilles reflex, biceps, brachioradialis. We do a whole bunch of ones just for fun in class. But I only ever test on these two. These two are the two that we're focusing on. The knee-jerk, aka patellar tendon reflex, and then the plantar reflex. So the knee-jerk reflex is the reflex that you see in movies all the time that we're all familiar with, where the doctor takes this little hammer and he hits your patellar tendon and your foot goes forward. The plantar reflex, what you do is, it's kind of demonstrated in these two photos. You take the handle, the side of the handle of your reflex hammer, or some sort of slightly sharp object, like a paper clip would probably work well, and you kind of rub it down the side of the foot and then across the balls of the feet, right through here. And you're looking for something called the Babinski sign. The Babinski sign is when people's toes kind of splay. They kind of separate and bend backwards. That's called the Babinski sign. And you should not see this. You should not see this. It's not a normal response for an adult. A normal response for an adult is they kind of curl their toes down. They curl their toes down. So you should know what an electroencephalogram is. An electroencephalogram is an electric brain recording. You are measuring the electrical activity of the brain. And just like with the electromyogram, you're just measuring all electrical activity below the pads. Now, there's not really much muscle activity below your pads when you put them on top of your skull. So it's mainly going to be brain activity. It's mainly going to be brain cortex activity, more close to the surface of the brain. We're kind of eavesdropping on the conversation of the cerebral cortex interneurons. An interneuron is a neuron in your central nervous system that talks to other neurons in your central nervous system. All electrical activity. It's not just going to be action potentials. Any electrical activity that happens below, we're going to pick up. So we're going to pick up graded potentials. EPSP are excitatory postsynaptic potentials. IPSP are inhibitory postsynaptic potentials. And we're going to get into these later on, but when nerves are famous for sending these electrical signals down their axons called action potentials. In the cell body, the neuron body, there are other local electrical activities that can happen. The action potential happens when the potential of the neuron increases to negative 55 millivolts. It's normally at negative 70 millivolts. And there's these local events, these local electrical events that happen on the neuron called excitatory postsynaptic potentials and inhibitory postsynaptic potentials. We're going to cover these later on in this lecture, and we're going to also cover them in the nervous system lecture. So we'll get to those in a minute. And action potentials create potential or electrical differences that are detectable with the electrodes. So let's take a look at this picture over here, because I mentioned this picture a few times throughout the semester in different lectures. So this ball that you see down here, that is a neuron cell body. A neuron cell body is the postsynaptic neuron. It's a neuron that is receiving signals from other axons, from other nerves. All of these things on top of this cell body that you see, all of those are neuron axons. Those are all axons that are forming connections with this cell body. There's a lot of them. 1, 2, 3, 4, 5. There's a lot of these axons forming connections with this cell body. The cells that are sending the signals are the presynaptic neurons. The cell that is receiving the signals is the postsynaptic. I just think it's really amazing to see what it actually looks like, to see those connections and see how many different neurons can connect with one neuron, how many axons can connect with one neuron. So electroencephalogram, so we have our data right here. Now we don't use this many electrodes in lab, we use like 3. But over here, you can see what the alpha, beta, delta, and theta waves look like. They each have a characteristic frequency. Now that frequency can change a little bit between person for different reasons. But throughout this experiment, the main thing that's going to change when people open and close their eyes and do the Jurassic maneuver is the amplitude. The amplitude is going to be how high are the peaks and how low are the valleys. So frequency, time from peak to peak. We have different characteristic patterns for each one. I want you to be aware of the frequencies for the exam. Amplitude is the distance from a peak to a valley. Peak to a valley, the activity / intensity. It's influenced by activity. No amplitude. So let's do a thought experiment. If there was no electrical activity happening in somebody's brain, what would be going on with that person? 0 electrical activity happening with that person. They would not be alive. They would not be alive. When we talk about the applications of this, we'll get to that a little bit also. If it acts kind of chaotically, if it acts really, really chaotically, it can be indicative of certain types of epilepsy. And when we get into the applications, we'll talk a little bit about that also. So I want you to know about the different waves and when each wave is kind of enhanced. So alpha waves, when you're awake and relaxed with your eyes closed, meditating, that's when alpha waves are have the biggest amplitude. Now they're called alpha waves because they were the first discovered. So when you meditate, when you have your eyes closed and you're relaxed but you're awake, your alpha waves have the largest increase in amplitude. Beta are invoked by visual stimuli and mental activity. So when your eyes are open and you're looking around, beta waves increase in amplitude. Delta waves are enhanced and increased amplitude prominent with newborns and sleeping adults. Sometimes they're enhanced with brain damage. And theta waves are enhanced in newborns, sleeping adults, and people with emotional stress. Now, what's going to happen when you clasp your hands and pull apart, aka perform the Jurassic maneuver? Perform the Jurassic maneuver. The Jurassic maneuver is going to increase the amplitude of all. So we're going to do the Jurassic maneuver in this lab and in the Reflex Lab. The Jurassic maneuver basically just increases brain activity. You clasp your hands together, you pull apart, and it's called cortical irradiation. It's going to increase the amount of activity happening in your cortex. and that's going to increase the amplitude of all the different waves. So before we, let's start at the basics. So let's start at the basics for the reflex. So I point this out in the lecture when I talk about this. This material right here is in lecture and lab, so fair game for both tests, laboratory quiz and exam, lecture exam. Do not confuse reflex characteristics with the components of a reflex. There's 5 characteristics and there's 5 components. So just giving you a heads up, students mess that up pretty often. So the characteristics of a reflex are they're involuntary. When the doctor hits your patellar tendon with the hammer, you don't try to move your leg. You don't try to activate your quadriceps. It just happens. It's involuntary. It requires adequate stimulus. The doctor uses a hammer and not a feather for a reason. He needs to have an adequate stimulus. There's a specific receptor. This hammer, what it does is it stretches your patellar tendon. It is a specific receptor within this tendon. It is a stretch receptor that activates this reflex. It's patterned or stereotyped. You get the same response every time. Your quadriceps contract every time with the knee-jerk reflex. It's graded, meaning as long as you have adequate stimulus, if you increase the force you do it with, you'll increase the response. So a medium strength hit will get a smaller response than a large strength hit of the hammer. Here is the other aspect, the reflex arc. So we have the receptor, which detects the stimulus. We have the sensory fiber, which relays the information. We have the integrator, which processes the information, the central nervous system. We then have the motor or efferent fiber, which carries that information to the effector, which causes change, affects change. So with this knee-jerk reflex, our receptor is going to be a stretch receptor within the tendon. It's going to send sensory information via an afferent nerve to our central nervous system. Our central nervous system is the processing center, and then the motor neuron is going to be our efferent nerve, our effector, which sends a message to our quadriceps to contract. So the knee-jerk reflex is considered monosynaptic. Monosynaptic because this sensory nerve in your spinal cord connects directly to the motor neuron. The information never travels up. So the information doesn't travel up your spinal cord. Now, after it happens, information will travel up your spinal cord telling your brain what happened. But the most direct pathway doesn't travel up your spinal cord. It goes sensory neuron directly synapsing on a motor neuron. Right there, the quadriceps. Monosynaptic. So the stretch receptor is the muscle spindle. Stretch receptor is the muscle spindle. It's kind of like the inside of a pen where you have this spring that's wrapped around the ink tube. It's like you've got these nerve fiber endings like wrapped around this little spindle, and it detects stretch. So that is our specific receptor. That is our specific receptor. The stretch receptor in muscle spindle. So when you stretch this tendon, when you stretch it, when you stretch this spindle, this muscle spindle right here, these coils, we stretch them, it's going to cause sodium ion channels to open. those sodium ion channels are going to open and sodium is going to rush into this nerve cell. Now, nerve cells are normally at a resting negative 70 millivolts. Resting negative 70 millivolts. When sodium rushes in, you are adding positive charges to the cell. So when you add positive charges to the cell, it's going to increase. It's going to increase. When you stop stretching the muscle spindle, those channels are going to close and you're going to go back to that resting state. If the stimulus is strong enough, if enough sodium rushes into the cell to cause the cell to depolarize to negative 55 millivolts, the signal will be sent. I'm going to repeat that. If enough sodium rushes into the neuron that it depolarizes to negative 55 millivolts, the action potential will be sent, the reflex will be initiated. That is adequate stimulus right there. That is adequate stimulus. And that those ions rushing in is an excitatory postsynaptic potential. It's a graded potential. It is a graded potential, a local change in electrical activity within the neuron body. So deep graded potential in the sensory neuron. The graded potentials are caused by ion channels opening on dendrites and cell bodies, and ions flowing in or flowing out and changing the polarity of the cell in that local cell body area. So, stretch right here. Actually, we're going to move on. All right, so when enough sodium rushes in, you have an adequate stimulus, that enough sodium rushes in, that it depolarizes the neuron to negative 55, you will have your action potential, you will have your signal sent to the central nervous system. It's going to carry that signal to the spinal cord. That's the action potential. So action potential is that electrical activity down the axon that's going to travel and send the message long distance. Graded potential is within that cell, within that local area. Within that local area, sodium ions rush in, depolarizes. So right here, this is just showing that electrical activity being sent down the axon. We're going to get into exactly how that happens in the nervous system lecture. Perfect. So once that signal, once that action potential gets down here to this motor neuron, it's going to send neurotransmitters across the synapse, which are going to connect, which are going to bind to receptors on the motor neuron. So right here we have the presynaptic sensory neuron. When that action potential comes down the axon, it's going to cause the release of neurotransmitters into the synapse right here. When those neurotransmitters enter the synapse, they're going to bind onto the receptors on the postsynaptic cell. When those neurotransmitters bind onto the receptors in the postsynaptic cell, they're going to open up ion channels, sodium ion channels. It's going to allow sodium to rush in again. And we're going to get another graded potential, another local change in the potential of this postsynaptic cell. The motor neuron is just like the sensory neuron in the sense that it's at rest at negative 70 millivolts. When enough sodium rushes in, that it depolarizes to -55, the motor neuron is going to send the action potential down the axon. And it's going to cause the muscle to contract. Perfect. So the sensory neurotransmitters that are released by the axon are going to cause graded potentials in the motor neuron, which is going to depolarize the motor neuron enough to send an action potential back to the quadriceps. Action potential in the motor neuron is going to travel from the spinal cord all the way to the muscle. Perfect. Perfect. Causing the muscle to have a response. All right, so we followed this pathway right through here so far. Sensory neuron synapsing directly with motor neuron to the quadriceps. But you might notice that there is a fork in the road, that this sensory neuron also synapses with this interneuron, And then this interneuron synapses with a motor neuron that goes to the hamstring muscle. This is polysynaptic right here, because there's many synapses. What this is doing right here, what these interneurons are doing right here, is these interneurons are inhibiting the hamstring muscle motor neuron. So the signal is sent down the sensory neuron to the central nervous system. The sensory neuron synapses directly with the quadriceps motor neuron. It's going to connect to some interneurons, and those interneurons are going to decrease the likelihood that the hamstring muscle is going to activate. These interneurons are going to turn off the motor neuron for the hamstring muscle. Now, your body wants to activate the quadriceps to have your foot go forward. If the hamstrings are activated, that's going to prevent that from happening. So it makes sense that there's the reflex also turns off the hamstring muscle. So an EPSP is an excitatory postsynaptic potential. It's a graded potential on the cell body of a neuron that increases the likelihood that it depolarizes to that -55. We see the EPSP right here where the sensory neuron synapses directly with the quadriceps motor neuron. The opposite is going to happen when this interneuron synapses with the hamstring muscle. This interneuron wants to inhibit, it wants to prevent the hamstring muscle from sending the signal. So it's going to cause an inhibitory postsynaptic potential. It's going to cause this motor neuron, this hamstring motor neuron, it's resting at -70 millivolts. It's going to move that potential away from -55. So instead of sodium rushing in and causing it to move towards -55, potassium is going to rush out. You're going to remove positive potassium ions from this motor neuron, which is going to cause it to go from -70 to lower, like -80 millivolts, which is going to be inhibitory. It's going to decrease the likelihood that this motor neuron sends its action potential. So IPSP, inhibitory postsynaptic potential, prevents simultaneous quadriceps and hamstring contraction. So quadriceps motor neuron, excitatory, sodium rushes in, it depolarizes it, it causes an action potential. Hamstring muscle connected to interneuron, inhibitory postsynaptic potential, decreases the likelihood of an action potential to happen. Potassium rushes out, causes the cell to be more negative. When performing the gymnastic maneuver, what happened to the knee-jerk reaction? So we're going to do the knee-jerk reaction, knee-jerk reflex, or the patellar reflex, just when the students are sitting there. And then we're going to do it again when they're performing the gymnastic maneuver. Do you think that's going to have an effect on the reflex? Well, it actually is. It actually is. But when you do the Gendrastic maneuver, you increase the effects of the knee-jerk reaction. You get a larger response, the foot moves forward more when you're doing the Gendrastic maneuver while you hit the patellar tendon. And you do this because of something called Renshaw cells. You do this because of something called Renshaw cells. So let's follow this pathway. We've got the receptor, which causes an EPSP in the sensory neuron, which sends the action potential to the motor neuron, which causes an EPSP in the motor neuron, which causes a muscle to be sent to the muscle. The motor neuron causes a signal to be sent to the muscle. In your brain, you send signals to something called a Renshaw cell. So normally, our brain wants to kind of attenuate this reflex. Normally, our brain wants to kind of minimize the effects of this reflex, wants it to have a reasonable response. So it sends signals to something called Renshaw cells, and Renshaw cells also synapse onto the quadriceps motor neuron. So both the sensory nerve and the Renshaw cells synapse onto this motor neuron. And the Renshaw cells are sending inhibitory postsynaptic potentials to your motor neuron. So sometimes the sensory neuron can cause an excitatory postsynaptic potential that's large enough that it can overcome the inhibitory potentials of the Renshaw cells, but it's still going to dampen, it's going to minimize the effects of the knee-jerk reaction. When you do the Gendrastic maneuver, you turn off the Renshaw cells. When you do the Gendrastic maneuver, you turn off these Renshaw cells. And so you turn off the IPSPs that are also synapsing with the quadriceps motor neuron. The Gendrastic maneuver distracts interneurons. It causes cortical irradiation, which removes the IPSP. Now, this word is a double negative, which kind of can cause some issues with some students. Disinhibition. Disinhibition is what it's called when you remove the Renshaw cells from functioning. So normally the Renshaw cells are inhibiting the motor neuron. With the Gendrastic maneuver, you disinhibit, you remove the inhibition, allowing for a larger knee-jerk response. Disinhibition. Perfect. So this all happens at the L4 spinal cord. So the Renshaw cells are connected to your brain. The Renshaw cells, their pathway connects all the way up to the brain. This sensory neuron to quadriceps is all at the level of L4, the L4 spinal nerve. Sensory information goes in L4 and comes out L4. The Renshaw cells are connected up to the brain. Let's look at the plantar reflex now, which is considered to be polysynaptic. There's many synapses. With the plantar reflex, we kind of brush along the bottom of the foot. Normal response, toes kind of curl down. The Babinski sign is when the toes flail up. The pathway for this reflex, you have the receptors in your foot. It relays the sensory information to your spinal cord, the L5-S1 level of your spinal cord, and then that information goes up the pyramidal motor cortical spinal tract. Basically goes up your spinal cord to your brain, and then back down your spinal cord to your motor neurons and to your feet. So knee-jerk reaction, sensory nerve to L4 spinal nerve to spinal cord, same level back out, doesn't go up to the brain. Renshaw cells come down from the brain to down your spinal cord. Plantar reflex starts at the foot, goes up sensory neurons to the bottom of your spinal cord, goes up your spinal cord to your brain, back down to L5S1, and then out the motor neurons there to your feet. In infants, this can be normal. This is normal in infants. The Babinski sign. The Babinski sign is normal in infants, the toe flailing. Adults, it's not normal. There's something damaged along that pathway if you get the Babinski sign in an adult. It could be anywhere along that pathway, but something along that pathway, from receptor to sensory nerve to L5S1, up the spinal cord to the brain and back down. Anything from a brain tumor, which would be the brain level, a slipped disc, which would be the nerve level, spinal nerve level, anywhere along that tract. Now, knowing what you know now, what would the following types of spinal cord damage, how would it influence these two reflexes? So this is getting a little bit away from physiology and more into pathology and diagnosis, but let's apply the knowledge that we have. For your knee-jerk reaction, if you have damage below the L4 spinal nerve in your spinal cord, how would it affect the knee-jerk reaction? How would damage below L4 in your spinal cord affect the knee-jerk reflex? How would it affect it? What about damage to your cervical spine? If you have damage to your spinal cord at your cervical level, how would it affect the knee-jerk reflex? Same with the plantar reflex, same with the cervical. So same with your plantar reflex for damage below L4 and damage in your neck. Does the knee-jerk reaction, does that pathway ever go to the spinal cord below L4? How would it change? Does the knee-jerk reaction, what components of the knee-jerk reaction travel through the spinal cord at the level of the cervical spine? Your plantar reflex, does that pathway involve the spinal cord below L4? Plantar reflex, does that reflex, what components of it, how does it utilize your spinal cord at your cervical neck level? And how would that affect? So knee-jerk reaction below L4, that pathway never goes to the spinal cord below L4. Damage to your spinal cord at the level of L5 or S1, normal knee-jerk reaction, normal re-jerk reaction. That pathway never goes to that area. Knee-jerk reaction for damage to the cervical spine. Well, those Renshaw cells are receiving messages from the brain. Damage to your cervical spine, to the spinal cord at your cervical level, could affect those Renshaw cells. If those Renshaw cells are permanently turned off because of damage to your spinal cord, well, it can have an enhanced knee jerk. It's like doing the Gendrastic maneuver without doing the Gendrastic maneuver because you're turning off the Renshaw cells, you get an enhanced response. Plantar reflex goes into the spinal cord at L5S1, damage, it's below L4, that's can cause the Babinski sign. Damage anywhere along the pathway causes the Babinski sign. The information for your plantar reflex, it travels up your spinal cord to your brain, it goes through the cervical spine, Babinski sign if there's damage in the cervical spine. All right, if you have any questions, please let me know, and I will see you in the next video.